you, and kia ora koutou. Yes, thank you for the opportunity to share with you some of the work that we've been doing on the invasion of the common myna in New Zealand, particularly. I'm Annabel Whibley. I'm based at the University of Auckland. This is Marsden-funded work led by Anna Santure at the University of Auckland as well, and with a number of collaborative partners, including Lee Rollins at the University of New South Wales, who you heard from this morning, Richard Major and Rebecca Joihnson, who worked at the Australian Museum at the time. Rebecca's now at the Smithsonian, and with contributions from a number of other people that I'm going to talk about. But I do want to acknowledge Mark Peck from the Royal Ontario Museum, who's provided us with a lot of historical specimens, and very many people from around New Zealand who've given us contemporary samples, without which we couldn't have got very far. Also acknowledgements to Nessie, who support all our HPC work, and genomics Atsawawaranunt, who funds some of my other projects. So I'll skip through the introductions to a large extent because this is preaching to the choir, but essentially there's a huge imperative to work on invasive species. They cause immense ecosystem and economic harm, and there's increasing impetus to do this because this threat is increasing with global trade and mobility trends and pressures coming from climate change events. And genomics can inform what we do in the invasion space in a number of different ways. It can provide insights into invasion sources, dispersal properties, other demographic parameters, a sense of what the diversity is, what adaptive potential you have in your invasive species, and many other things. And I guess there are applied and fundamental goals within this space, and we heard quite a lot yesterday about some of the kind of intervention and management and surveillance strategies we might use. I just want to go off topic for a moment and share this one with you, because this one I hadn't seen before as an intervention, but it's basically, what is it? Fighting invasive species one bite at a time. And the idea that you might, as a way of control, start to eat your invasive species. But that's not what we're doing with myna. I'm not advocating that at all. So with myna, we're really looking at a system that we can use to try and get into some of the fundamental properties about invasion. And we've heard again a lot about the genetic invasive species paradox and trying to unpack that a little bit and looking mechanistically at mechanisms of rapid adaptive evolution. And I'm particularly interested in the idea of the constraints that the genome might offer up kind of intrinsically due to its organization and other factors within it. And what I'm going to talk about today is really an overview of the project that's going on at the moment. I'm going to start by talking about what we know about the population genomics of myna in New Zealand, a little bit about how we've gone about creating a high quality reference genome. And then the next bit's a teaser, and it's a teaser basically because we haven't finished yet. But we really want to look at structural variants and transposable elements and evaluate them as agents of rapid adaptive evolution. So again, a kind of graph that you've seen, or map that you've seen a lot of today. This is the distribution of myna, the native ranges in blue. It's predominantly the Indian subcontinent and Southeast Asia. And then it's been introduced deliberately in most instances across a large part of the world, particularly to Pacific Ocean and Indian Ocean islands, to Australia and New Zealand. And these are largely separate introduction events, and where myna was brought in to control grain pests, and largely in the late 19th century in terms of time. And the jumping off point for our study was work conducted by Ewart, Richard Major at the Australian Museum, where they looked using DART-seq, so a form of reduced representation sequencing, at the population genomics of the myna invasion in Australia. And there's a lot to this paper, and I'm really just going to focus on the population structure side of it. So this is what's shown here. There are three clusters, three genomic clusters in Australia. And just to unpick that a little bit, we have introduction points to the Gold Coast, to Sydney and to Melbourne, and then a subsequent translocation event from Melbourne up to North Queensland. This is where myna is found in New Zealand. So it's largely confined to the north and central parts of the North Island. And we recently had a summer scholar, Anneke Beasley, who did some work looking at the historical introduction of myna. And so she was looking both at the academic literature, at occurrence reports, but also digging into kind of contemporary newspaper records, some of which are shown here, which chart the spread of myna across New Zealand, and looking at acclimatization society records and other kind of contemporary reports. So the earliest introductions were in the 1860s, 1870s into New Zealand. And this is kind of a summary of her findings, and shows the main introduction sites, or the introduction sites that she could find in New Zealand. So largely the agricultural centres, and quite interestingly, not at all where it's found today. So myna has effectively self-introduced into the North Island from these deliberate introductions into the South Island and south of the North Island. This shows how the distribution changed over time. And at this point, we were able to look with Dartseeker in New Zealand. And this is work that's been led by a PhD student in the group. And the panels on the right show his sampling design. So we have a lot of samples across New Zealand. These are largely contemporary samples. There are also some historical samples from the 1970s, 1980s. And those are also complemented by historical samples from the same time period from the native range. And so this really augments what Kyle was able to do, because he had just contemporary samples from Australia, but we were able to provide a little bit more context, as well as to start to look at New Zealand, but using the same methodology. This is the population structure in New Zealand. It's not desperately exciting, I guess, or at least it's quite clear cut. Essentially two clusters, one of which is most of New Zealand, and the other which is the Napier region in the east. And this makes a lot of geographical sense because there's this axial mountain range that looks to kind of limit dispersal. I'll note as well, though, that we could do with a better sampling around the limits of that range. And so if anybody knows anyone who's controlling myna in some of those regions, please encourage them to get in touch, because we would like to look more at the expansion fronts if we can. So A was also able to look at the distribution of, at the introduction history or the signatures of likely sources of myna. It's complicated, and it always is. So myna from New Zealand cluster with myna from Melbourne, which fits with our understanding. They also cluster with myna from Maharashtra in India, which again, so Maharashtra, the port of Mumbai, that was the origin, then into Melbourne, then into New Zealand. But unfortunately, Maharashtra has two distinct clusters, and so also, and we have no understanding of why we have that distinct clustering in Maharashtra, but there's always something that doesn't quite fit, right? And A's gone on to look at, for example, folded site frequency spectra. So here you can see the native range population on the left. You've got quite a preponderance of rare alleles, and there's progressive bottleneck as you go through. And populations in Hawaii and South Africa, which were founded by quite small numbers of individuals, are really quite severely bottlenecked. A's also delving into ecological niche modelling, and this is just a placeholder for that. So starting to understand how ecological and environmental factors affect the distribution of myna in its native range, how that corresponds to the parameters in New Zealand, and how that might change going forward. All of the population genomics work was done with a fairly rudimentary reference genome that we generated using 10x data. It's reasonably complete, but it was really fragmented. And so one of the things that we set out to do was improve upon that. By sheer accident, we generated two long read data sets, one PacBio, not hi-fi, just regular PacBio, and one nanoport long read, that are very, very similar in the statistics. So almost exactly the same amount of sequencing from both technologies. The PacBio came first. We weren't very happy with it. We got nanopore data much more recently. Interestingly, for those of you who are into genome assemblies, if you assemble them independently, they do quite differently, despite the fact that they're very similar. So our PacBio genome still stays in about 17,000 contigs and is quite incomplete, less than 80% buscos. The nanopore data assembles much more nicely, 96% complete, half the genome in scaffolds in 30 scaffolds. Really quite good stats for what is actually quite a low coverage 20x assembly. You can improve that a little bit if you put the two data sets together. And we're currently going through just the last stages of manual curation before we freeze this genome assembly. And generating a really high quality genome isn't important for all applications, but was something that we really wanted to do, because although we've talked a lot about SNP variation and structural variants as well, we particularly want to interrogate the repetitive content of the genome, which is essentially a subclass of the structural variants, but the hardest one, I think, really to get at. And so having a very high quality genome will really support that. This is work that's being led with Kat Stuart, who talked to you earlier about her PhD work on starlings. I guess I just really wanted to emphasize that TEs are really often overlooked in these kind of studies, but they're actually a really rich source of genetic variation. And it's kind of interesting because, so for example, in this study from Arabidopsis, which is not by Kat, but by a different Stuart, they they found that nearly 2/3 of TE insertion polymorphisms aren't actually in linkage disequilibrium with the adjacent SNPs. And so what they're doing is you're not capturing the information from the TE insertion polymorphism within your selection scans based on SNPs alone. And they can have really quite interesting effects on gene expression and particularly alter epigenetic states. And so the potential for TE transposition to catalyze quite rapid changes in the genome is maybe makes them quite interesting in the context of an invasive species having to potentially adapt to a new environment. There's not a lot of data out there in TE polymorphism in wild populations, but we can just look at our reference genome individual to get a little bit of a sense of the transposon element landscape, and that's what's shown here. And interestingly, for myna on the left, you see that spike of green very close to zero. That reflects a burst of relatively new transposable elements, so they've all got quite high identity. And so it suggests that there are active elements in the myna genome, which is really encouraging as we go on to look at this. It's shown in contrast to starling on the right, which has a little bit less going on in terms of recent transposition, which is maybe a little bit disappointing because starlings and myna are 2 of the 3 birds that feature on the IUCN Global Worst Alien Invading Species. And so what we really want to do is extend the analysis to starling as well. So we have a relatively small collection of starling, but we plan to do the same work in parallel. And to try and really understand whether there are any features of these genomes that kind of explain the invasive potential of these birds, and to put it into a much broader phylogenetic context. And I'll end there and just thank you and happy to take any questions.
Thank you so much. Does anybody have any questions?
Thanks. Excellent talk. So I am really interested in the role of TEs in invasion, and there is some, I don't know, maybe you're familiar with it, there's some work in plants also looking at this. And so, I don't know, like, what do you think the general pattern could be?
So if I'm brutally honest, I think it's much more compelling in plants. Plants do crazy things with their genomes. They have lots more transposable elements in the first place, but it's also very messy. The really nice thing about bird genomes is that they're really quite compact and quite conserved. They only have like typically between 7 and 15% repetitive content. And so we're actually quite well powered to resolve what's going on. But I think there's much less evidence, for example, for environmental stress really triggering bursts of TE movement in quite the same way in plants. But I think we don't really have the tools to do this as well as we might like to. But yeah, I think it's really fascinating.
All right, we should talk more about it.
Anybody else?
I'm just curious about your collection locations from Maharashtra. And I'm wondering whether it's the same thing as the cane toads with coastal and forest.
Yeah, the problem is we just have no information, and we have no way to get that information. So it's, I think it's one of those mysteries that we feel like we're never going to get to the bottom of. We can do due diligence on like other potential confounding factors. We can re-extract and test the samples again and just make sure there's nothing like that. But we just have the museum metadata, and it's not very informative, unfortunately, for those. It's also very hard to get samples from India, contemporary samples.
This might be just naive of me, but I was quite surprised to see the outperformance in the genome assembly of, nanopore over PacBio. What do you think explains that?
Bear in mind, it's not hi-fi, it's regular PacBio, but I suspect that it's a problematic library. I think it's very likely a one-off. I certainly wouldn't expect that for hi-fi data, and I was very surprised to see it. Like, we knew already that the PacBio dataset wasn't very good because it wasn't very good in scaffolding, but to set them aside side by side, because their length distributions are actually very similar. It's not like there's an increase in long reads in the nanopore data. It's a museum specimen, and we're really limited by its properties, so we don't have much stuff beyond 50 kb.